Hello students, good morning, Jai Swaminarayan. In previous lecture, we have discussed about principles of inheritance and variation, in which we discussed a working method of Mendel's and their principles, in which we discussed about two principles. First principle is a law of segregation and another one is a law of dominance, which is mainly based on monohybrid cross, where only one character is taken as a study in studying. Now today we will discuss about dihybrid cross and another principle of inheritance on the basis of dihybrid cross, which is a law of independent assortment. Before we starting today's lecture, let's summarize what we learned in previous lecture. We all know that in a monohybrid cross we get 3:5 ratio, and a genotypic ratio is 1:2:1. Here 3:1 means out of 4 flower or P, 3 flowers are tall plants and 1 plants are dwarf. And the genotypes there are 3 types of genotype we get in a F2 generation, which is capital T, small t and small t. Here, on the basis of this genotype, we can determine the phenotype of particular plant. Like this T, capital T and capital T are responsible for formation of tall plant, and this small t is responsible for formation of dwarf plant. And in hybrid condition, capital T and small t, it is responsible for formation of tall plant. Now let's see, on the basis of phenotype, can we determine the genotype of particular plant? So let's try. This is the phenotype, tall and dwarf. If the plant is dwarf, we can say that the genotype of that particular plant should be small t. But the problems comes in a tall plant or in a dominant character, where there is 2 genotype and only 1 phenotype. So for tall plant, out of 2 genotypes, anyone can be present in a dominant, in a phenotypic tall plant. Now let's see on the, so how can we determine that in tall plant which genotype is present? For this, Mendel developed one cross and given the name that test cross. In test cross, we cross the dominant character with the recessive character to get to know that the parents or particular unknown dominant is a heterozygous or homozygous. Means it is a homozygous having both the similar allele, or the genotype is a heterozygous, means both the alleles are dissimilar. So let's see what is the test cross. Test cross is the cross between dominant trait and with the recessive trait to know whether dominant is a homozygous or heterozygous. Let's take one example. We have one plant with the dominant character violet color. In this plant, all the flowers are violet in color, which is dominant on white color's flower. but we don't know the genotype of particular individual. There can be two genotypes. One is homozygous or another is heterozygous. Here capital T and capital T and small t. This genotype is known as a heterozygous, and this genotype is known as homozygous. Because this genotype have both the similar allele, where this genotype have both the dissimilar allele. Now let's see how we can determine the genotype or homozygous or heterozygous on the basis of dominant character or phenotype. The violet color flower or dominant character cross with recessive character. Here white is a recessive character. Here both are small w, which stand for recessive trait white. This dominant is cross with recessive allele white. So let's see, there can be two possibilities in dominant character. One is a hybrid or heterozygous, where another one can be homozygous. Let's see if the plant is a heterozygous and it cross with white recessive, what will happen? In white only it is a homozygous. That's the reason only one kind of gametes will form, and that is a small w and white trait. On other hand, this dominant have two different kinds of allele, and it is a heterozygous condition. That's why there are two different kind of gametes will form. One will have trait of violet color, which is symbolized in capital W, and another 50% gametes have white color flower trait. Let's cross between them. In zygotic condition, we will get two different kinds of flowers. 50% flower will have violet color and 50% flowers have white color. Let's see here. This is the capital W and this is the small w. Here what we do, that first we will write this w and then we will write this small w. What we will get here? Heterozygous dominant, which is responsible for formation of violet color flowers, and in another here is a small w and here also a small w. So here zygote will form with the both the allele having small w having a white allele. Both the alleles will have white trait. So the zygote will form or the plant will form which have both the white allele. That's the reason he will produce into a white flower. So if the dominant character is heterozygous in condition, in that condition we will get 50% white flower and 50% violet flower. Let's see the another scenario where dominant is homozygous in condition, where both the alleles are dominant in nature and which is stand for a violet color. Let's see here. Here it is present in homozygous condition. That's why he will produce, or it is produced, only one kind of gametes here, and this is also present in a homozygous condition. That's why this is also produce one kind of gametes, and in the fusion of gamete, at the fusion of gamete, it will produce heterozygous dominant, as you can see here, which is responsible for formation of violet flower. If now let's see here, if the dominant character have heterozygous, in heterozygous condition, it will produce half of the violet plants and half of the white plant. On other hand, if the dominant having both this in a homozygous condition, it will produce all the violet plants in a test cross. Now one IMP question can be asked in exam, that in test, what is the result of test cross in a dominant heterozygous condition? So in heterozygotic condition, test cross result is always in 1:1. Okay, so this is all about test cross. Now we will see and discuss about dihybrid cross, and on the basis of dihybrid cross, one principle is there which is known as principle of independent assortment. So let us see dihybrid cross and principle of independent assortment. So the next point is the limitations of Mentalism. There are certain limitations of Mentalism, in which we will discuss about 3 of them. First one is incomplete dominance. The word itself says everything, that in incomplete dominance, the dominant allele is not capable to dominate on a recessive allele. Let's see what is the incomplete dominance. It is a phenomenon of neither of two alleles being dominant, so the hybrid is fine mixture between them, which means in incomplete dominance, the dominant out of both the alleles, neither alleles are capable to dominate, and the offspring will produce is a hybrid of both the traits. The best example of the incomplete dominance is Mirabilis jalapa, in which there are three different color of flowers are occur. First one is a red flower, pink flower, and white flower, in which here RR is a RR is for red flower and small RR is for white flower. When cross between them, the hybrid from is a pink flower. अगर वो मेंडलिज्म को फॉलो करता होगा तो यहाँ पर आर की जगह पर क्या आएगा? इट इज हाइब्रिड इन नेचर. अगर मेंडलिज्म को फॉलो करता होगा तो यहाँ पर आर हाइब्रिड है तो भी डोमिनेंट की वजह से रेड फ्लावर मिलेंगे, पर यहाँ पर मेंडलिज्म को फॉलो नहीं करते हैं, उसी की वजह से यहाँ हाइब्रिड में पिंक फ्लावर मिलेंगे, जो कि मिक्सचर ऑफ वाइट एंड रेड फ्लावर. ओके, वाइट में से थोड़ा और पिंक में से थोड़ा तो मिलके बनेगा पिंक फ्लावर. एंड इन F2 generation, when we allow to self pollinate this pink flower, they will produce 3 different kinds of flower in F2 generation in ratio of 1:2:1. One flower of red flower, one plant with the pink flower, and two plants with the pink flower, and one plant with the white flower. So one important question can be asked in exam, that what is the genotypic and phenotypic ratio occur in incomplete dominance. So the answer is 1:2:1 in both genotypic as well as in phenotypic ratio. Now next is codominance. Codominance is the phenomenon of two alleles lacking dominance and recessive relationship, and in hybrid condition both the alleles are separately present on the hybrid in a hybrid offspring. The best example of codominance is human blood group. In codominance, the phenomenon of two alleles lacking dominance and recessive relationship, and both express themselves in offspring. For example, let's see here. The I gene is responsible for blood group in human. Okay, there are three types of blood group: A, B and C. No, there is four type of blood groups. There is A blood group, B blood group, AB and O blood group. This is all controlled by three alleles. Name of this allele are IA, IB and IGO. In blood group, person having blood group A will have alleles, either he is homozygous or heterozygous. Likewise, in blood group B also allele B in a homozygous and in a heterozygous. When both the alleles are present, the offspring will have AB blood group, which suggests that they are expressing themselves in a hybrid condition. And next one is multiple alleles. Multiple alleles is more than two alleles are control the particular character or trait known as a multiple allele. The human blood group is also one of the most very good example of the multiple allele, in which three alleles are responsible for human blood group. Okay, so today we will discuss. आज हमने क्या देखा? तो आज हमने dihybrid cross and on the basis of dihybrid cross, one principle, that is independent assortment, and then later we seen three different limitations of Mentalism, in which we discussed incomplete dominance, codominance and multiple alleles. In next class we will discuss about linkage and recombination, and then we will discuss about certain genetic disorder. So this is all about today. So next point is the inheritance of two genes. The inheritance of two genes means the cross made to study inheritance of two Mendelian factors or gene, known as inheritance of two genes, which is also known as dihybrid cross, which means to study the cross to study the two characters with in a same plant is known as dihybrid cross. On the basis of dihybrid cross, Mendel gives a new principle known as laws of independent assortment. The laws state that when two pair of traits are combined in a hybrid, the segregation of one pair is independent to another pair. The allele of two traits are segregated during gamete formation from independent to each other. For example, Let's take one character, tall and dwarf plant, and another character is round seed and wrinkle seed. So the allele for both the traits will segregate independently during gamete formation, and these alleles are rearranged during fertilization, and from two new different kind of combinations in trait or character. This law is known as independent assortment, which simply state that the allele for two characters or two traits segregate independently to each other during gamete formation, and during fertilization they rearrange in new combinations. This law known as independent assortment. Let's see in the dihybrid cross. What Mendel does in dihybrid cross, he take one plant जिसमें येलो एंड राउंड सीड थे. कैरेक्टर्स ऑफ सीड इज राउंड एंड येलो which is dominant over the seed, which the seeds are wrinkle and green. The genotypes of round and yellow seeds are capital R, and capital Y, crossed with the recessive trait wrinkle and green, having genotype of small r, and small y. During gamete formation, due to this is in a homozygotic condition, they will produce only one kind of gametes with capital R and capital Y. One allele for round and one allele for yellow trait, and both are dominant in nature. On other hand, in female or another plant, one allele from wrinkle trait and one allele from green trait will come in a gamete. Here also, due to these traits are present in a homozygotic condition, they will produce only one kind of gametes, and after fertilization of both the gametes, they will produce one hybrid plant which is similar to one of the dominant parent, having hybrid genotype or heterozygotic offspring, heterozygotic hybrid. When Mendel allowed them to self-pollinate, this F1 generation will produce different kinds of gametes, because here it is present in a heterozygotic condition. That's why he will produce, or it is produced for different kinds of gametes. Let's see what are the combinations of these gametes. First one will R bind with this Y and R Y, then it is with new combination with a small I, small y, then capital R and small y. Here one allele of dominant R and one allele from recessive y. Likewise, small r with the capital Y and small r with the small y. Here small r and small y is both are recessive in character, recessive in nature. After F1 generation, Mendel allow them to self-pollinate and self-breed. As a result of this, in F2 generation, there are 4 different kinds of combinations occur, which are 9:3:3:1 and 16 genotypes. In F2 generation, remember that in F2 generation, in dihybrid cross, in F2 generation, 16 type of genotypes are formed and 4 type of phenotype are formed, which are out of 16, 9 plants are round yellow, 3 plants are round green, where one is a dominant, round is a dominant character and one is a recessive character. Third one is out of 60 plant, 3 plant will have wrinkle and yellow, and out of 60 only 1 plant will form of wrinkle and green seed. My dear student, don't be confused with this number. These are the ratio of 9:3:3:1. This is not an actual number of plants Mendel get at the time of F2 generation. This is the ratio. The number of plants can be variable, but they are present in this ratio 9:3:3:1 ratio. And genotype, there are 16 genotype will from in F2 generation. Plants number can be variable. So this is all about dihybrid cross. Now we will discuss about certain limitations of Mendel. Yes, all things have certain limitations and certain exceptions. In Mendelian genetics, there are certain exceptions are also present. For example, certain genes are there who don't follow the Mendelian laws and Mendelian genetics, and these genes and these condition known as exception in Mendelism. There are these exceptions occur due to interaction between genes and interaction in the genes. This occur due to 2 types of interaction. First interaction is intragenic interaction, and another is intergenic interaction, in which in intragenic interaction the interaction occur in a gene, in a particular gene. For example, this is gene. This is one gene. The interaction occur in this gene known as intrageneric interaction, and the interaction between 2 different genes known as intergeneric interaction. Here intrageneric here is intragenic and intergenic combination. So let us start to different let's see the different situation where Mendelian genetics, where gene doesn't follow the Mendelian genetics.